Navya, a second year student at Azim Premji University, Bangalore. I am a biology student, as you can tell by the picture of larvae that's clearly over here. And the project I'm going to be talking about explores how chemical dyes, specifically a chemical dye called tartrazine, can be used to achieve and optimize optical transparency in fruit fly larvae. Alright, take a moment. Picture any scientific progress you can think of. It can be a Nobel Prize winning discovery. It can also be that one experiment that's been keeping you up for 3 weeks that finally worked out. Outside of realms of pure theory and mathematics, what is the most important part of any work that led to this progress? Collection of data. And this data is often collected in the form of images. Whether it is the X-ray that you take to figure out whether you've broken a bone, or CCD images used to analyze celestial objects, everything that we do requires images. We as a society rely heavily on images. However, in spite of all this, imaging techniques for medical science and life science research have significant drawbacks associated with them. MRI machines, which are used to perform anatomical studies, cost up to $1.2 million. And repeated exposure to radiation, such as in X-rays, can have cumulative dosage risks for groups such as children or people with chronic diseases. And lower quality of data obtained can lead to things like misdiagnoses of patients or incorrect inferences made during research. So, we as a society need to improve our imaging techniques for medicinal research and for life science research. The main things that we need to focus on is that this imaging technique should be non-destructive, non-invasive, and cost-effective for everybody involved. One step in this direction was conducted by O et al. In a Stanford laboratory, where they basically used a chemical dye called tartrazine and experimented on mice tissue and on chicken breast tissue to make the outer layers of the organism transparent. So essentially, you can see through the organism and get real-time data and perform more like volumetric analysis as well. So, O et al. Conducted a study on achieving optical transparency in living tissue using the chemical tartrazine. Is this a laser as well? Can I use this to point?
No, I think so.
Alright.
There is a light on top there.
It does a button.
May not be.
May not be? Alright, no problem. So, they showed that adding molecules that have high absorption resonances in the blue and the UV ranges of the spectrum can often help improve optical transparency in wavelengths that we can see. So, tartrazine is one such molecule. It's a yellow colored azo dye that's used as a food dye in moderate concentrations. And it has very sharp absorption resonances in the blue and the UV range of the spectrum. This means that it is capable of reducing the refractive index contrast between water and lipids by increasing the refractive index of the aqueous medium around it. Therefore, this means that it improves optical transparency in nearby visible wavelengths. So that is the structure of a sodium salt of tartrazine, which is what was used for experimentation. And here are the results of the Stanford group with chicken breast tissue. As you can see, when the chicken breast was exposed to higher concentrations of tartrazine, it became more see-through. So the research questions that we wanted to answer with this project was to expose third instar Drosophila larvae to different concentrations of tartrazine and see how long they survive for. Because it's important that your organism does not die with your imaging technique, right? It needs to be a non-destructive technique. So we checked for survival, and we also checked for what concentration and what duration of exposure to tartrazine produces optimal translucency in third instar larvae. So you want to get the best results possible such that your organism survives and you are able to see clearly. So we performed 2 sets of experiments. The first was a positive test to see for ourselves whether O et al. Study works or doesn't. So we experimented on chicken breast tissue and we tested for both transparency and its relation to concentration and the reversibility of the effect. Because you want to make sure that your organism is able to survive and function normally and optimally even after imaging. And the paper had shown that this effect is reversible in chicken breast tissue. but we wanted to test for that as well. And the second bit, which is in my opinion the more interesting bit, is to optimize the conditions for optical transparency of fly larvae. So we tested for both transparency and different concentrations and durations of exposure, and for survival when exposed to different concentrations of tartrazine. So first was the positive test. We basically had 6 beakers of tartrazine solutions with 5 different concentrations and 1 control. The control was distilled water, and we kept concentrations of 0.3, 4, 5, 6 and 7 molar tartrazine. Then we took a piece of chicken breast tissue, divided it into roughly equal sized bits and placed 1 in each beaker for 45 minutes. Each piece was around 2 millimeters thick. So after 45 minutes, we took out the tissue and left it to blot dry for a little bit, and then placed it on top of a paper with a piece of writing on it. The more readable your writing is through the tissue, the better your results. As you can see, the control group is almost completely opaque. At 0.3 concentration, it's a little visible. At 0.4, the visibility is higher. 0.5, it's even higher, with the highest visibility being present at 0.7 molar concentration of tartrazine. This indicates a direct linear relationship between the concentration of tartrazine and the optical transparency you can see in chicken breast tissue. We also saw that the results were reversible. When you washed the tartrazine soaked tissue with running water, it turned opaque again. Though it took more water for the 0.7 molar concentration to turn back to completely opaque. Next, we experimented on Drosophila larvae. So, we used third instar larvae and divided it into 3 sets of 30 larvae each. So, we wanted to see what conditions for the larvae can give you optimum results. So, we tested for 2 separate conditions. One was the presence of yeast in the medium in which you keep your larvae. And the second was whether or not the cuticle of the larvae is degraded or not. Degradation of the cuticle was done using 10% N-heptane solution and 90% D plus limonene solution in a 1:9 ratio by volume. So we had 3 different sets. One batch was kept in a yeast medium and had its cuticle removed. One batch had its cuticle intact but was still kept in yeast medium. And one batch had the cuticle removed but was not kept in yeast medium. In the first set, we didn't get any results. The third set, everything died out, so it didn't matter whether we got results or not. Best results were seen in the second set. So our experimental procedure for this is quite similar to what we did for chicken tissue with respect to the exposure to tartrazine. We had 6 different beakers with 5 concentrations and 1 control, and we left 5 larvae in each beaker. However, the only difference was that in chicken breast tissue, we left everything for 45 minutes. However, here we picked out larvae, one at 5 minutes, 15 minutes, 30 minutes and 45 minutes. And then we imaged everything under red light and under blue light. Best results were seen under blue light. That's our setup. So that is the control. As you can see over here, 0.3 molar at 30 minutes had a good result. Then 0.4, one second, I'll just confirm this. Yeah. 0.3 at 30 minutes, 0.4 at 45 minutes, 0.5 at 5 minutes, 0.6 at 5 minutes and 0.7 at 15 minutes seem to have the most translucency. So this did not follow a clear mathematical pattern the same way the chicken breast tissue did, but it does show that this area of research is promising. There are results that are showing up. We also checked for survival of larvae after 45 minutes. In the control, 100% of larvae survived as expected. At 0.3 molar concentration, we had 3 live larvae and 2 pupae. In batch 3, which was your 0.4 molar concentration, there were 2 alive larvae, 1 pupa and 2 died. In 0.5 molar, there was 1 alive larva, 1 pupa and 3 of them died. 0.6 and 0.7, all the larvae died. So clearly these 2 concentrations can be eliminated for this study as there was no survival. So this was only a week-long project and therefore these are very preliminary results that we have gotten. So a lot more needs to be done to expand on this area of research. One possible method of further study is to image the larvae more systematically from the same view to see what structures are visible at what concentration. So look at it in a more detailed sense and see whether the same structures are visible at all concentrations. Next is to perform cytotoxicity assays to evaluate the toxicity of tartrazine at different concentrations. And the third is to test other routes of administration of tartrazine. Here we just kept it in the medium, but we could also test for routes such as topical application or direct injection. So all of these are very promising areas of study, and we hope to work on this further. This research was done and supported by the National Initiative on Undergraduate Sciences Biology Camp 2024, hosted at Homi Bhabha Center for Science Education, TIFR, Mumbai. Dr. Mayuri Rege and Uzma Sheikh from the Homi Bhabha Center for Science Education were our main project coordinators and guides throughout the process. I would like to thank them for their invaluable support. I would also like to acknowledge my fellow researchers and groupmates, Urvi Chakraborty, Swapnil Kumar Bhuyan, Swaran Ghoshal, and Suptik Roy. And I would like to thank Flame University for giving me the opportunity to present this work over here. These are the references. Thank you. Questions?
How do you feel presenting amongst undergraduate researchers who are predominantly presenting from social sciences and humanities? How do you feel making a presentation from pure
About
science?
something like this?
From pure sciences, yeah.
Studying science on its own as an isolated field is a redundant way of thinking because science is done by humans. You need to be able to introduce the humanity into everything that you do, right? Because everything that you do needs to have an application that is good for humanity, and it needs to be in such a manner that it's ethical, and you take into consideration the fact that we are all humans and all factors need to be considered. And also I think this research has applications for human development because tartrazine is an incredibly cheap compound in comparison to, say, getting an MRI machine, like an MRI scan from a hospital, or other methods of imaging that are maybe, they can introduce a radiation exposure, or they have lower quality of data, or there's a lot that is incorrect with the system that often leads to people paying high out-of-patient costs during checkups, or not getting checkups in the first place, leading to worse off conditions.
I think your answer reminds us about the importance of breaking some of these disciplinary boundaries and perhaps having greater conversations between social sciences, pure sciences, and so on.
Yeah.
So thank you for that answer. A question about your research. One of the dilemmas we're facing is that if you kind of increase the concentration of tartrazine, then you get better transparency,
Pure organisms are dying.
but toxicity is increasing, right? And you spoke a little bit in your future directions. Can you propose some ways in which we can perhaps balance this out in future research?
So the idea is that we want to test this across multiple organisms to see what is the range of toxicity in, say, Drosophila versus chicken versus mice.
Right.
And we want to see whether that range is consistent across, say, for example, vertebrates versus invertebrates, or if you say, for example, mammals versus reptiles versus birds. And we also want to see the structure of every organism is different when it comes to the outer layers of their skin, right? The concentration of lipid to water ratio will be different for everything. So we want to see at what setup can we get the highest transparency with the highest amount of survival. So first we need more data, but also I think it's important to model why tartrazine is toxic. So run, say for example, simulations where you see what exactly tartrazine does inside the cell that causes the cell to die. And therefore that will depend on, say, a small functional group of tartrazine that is, for instance, this is a hypothetical example. Let us say that it had a radical oxygen that is causing something in the cell membrane to burst. This is a hypothetical example, I'm not actually certain.
The question was also very hypothetical. So.
Okay, yeah. So, for example, it had a radical oxygen that's causing something in the cell membrane to rupture. We can maybe design a drug that is, the scaffolding is similar to tartrazine, but that radical oxygen is replaced with something that's maybe a little more safe for cells. That's also one way to look at it.
I also wanted to say the fact that you have a team from different universities is a really good sign in research. And I think we need to connect more across disciplines, departments, centers, and so
Absolutely.
on
Absolutely.
at all levels. So thank you for your presentation.
Very interesting. Thank you so much.
Thank you.